Hi everyone, my name is Casey Maguire. It's really good to be speaking with you today. First, I'd like to thank the organizers of this forum, especially Chris Coburn, for the invitation to speak today. I'm going to be telling you about a technology we've been working on for quite a while with my lab, and it takes a completely different approach than the majority of strategies for improving AAV vectors for gene therapy, such as capsid modification. This is my disclosure slide, which I'll leave up just for a second, but you can see it later if you need to. So very brief background in AAV-based gene therapy, as I'm sure many of you have heard of it. So scientists have been working on viral vectors for a long time, based on the fact that viruses can deliver their genetic payload very efficiently. So basically what's happened is we've engineered these viruses to carry healthy DNA to cells, such that you can correct a mutation and allow the cell to go from an unhealthy state to a healthy state. And it's a really exciting time to be in gene therapy right now. Again, as many of you know, there's 2 FDA-approved AAV-based medicines. One's Luxturna for blindness, and the other one is Zolgensma for spinal muscular atrophy. There's also a lot of clinical efficacy for multiple diseases, including hemophilia A and B. With that said, there's still some limitations to treat a wide variety of patients with AAV. First is many of us have pre-existing antibodies to the virus. So we've been exposed to wild-type AAV sometime in our life. We get these antibodies, and then those antibodies can completely block the vectors from delivering DNA to the proper cells. Another really unfortunate issue is in a few cases we've seen patients die at very high doses of AAV. And this is thought to be mediated by the innate immune system, perhaps complement, and it seems to be dose related. So having more efficient vectors is really important. Finally, transgene expression can wane over time, and I really need to give you a little background on that. So AAV is maintained outside of the chromosome. It doesn't integrate very efficiently. So if you give AAV to dividing cells or growing organs such as the liver at a very young age, and wait over time, you can see the transgene expression can go down because the virus is getting diluted from the cells as the organ grows. So why not just give another dose to the patient when they're older? Well, you've effectively immunized the patient on the first dose of AAV, so redosing currently isn't possible. I should mention that for all these 3 challenges, there's other strategies by other groups which are promising. However, not one of those can address all 3 of these challenges on its own. That's why what we're trying to do with our technology. So one of the first things you learn in virology is how to classify viruses. And one classification is whether they're enveloped or non-enveloped. So non-enveloped viruses, one example is AAV, is that they basically rupture the cell, lyse the cell and blow it up to release the non-enveloped virus to allow it to infect other cells. Sneakier viruses, such as the infamous coronavirus, basically take some of the host cell-derived membranes with them, which allows them to infect other cells. But in recent years, it's gotten a little murkier. So there's several examples of viruses that can come out of the cell as both non-enveloped and enveloped. One example is hepatitis A, and it's been shown this virus uses it for immunoevasion. And our lab was actually the first to discover that AAV vectors could actually be found inside these extracellular vesicles or exosomes. And we thought they might have some really unique properties. This is an example of an enveloped AAV. You can see the lipid around the particle, and the particle is shown with the arrow. So over the past few years, we've been looking to overcome some of these challenges with enveloped AAV. The first one we looked at was antibody evasion. So you can see a naked AAV on the left, which is susceptible to antibodies, and we imagined that the enveloped AAV would be shielded from these antibodies. And on the graph at the bottom, that's the IVIG that has just purified antibodies, which have a lot of anti-AAV antibodies. And you can see that the enveloped AAV is much more resistant to these antibodies than standard AAV. To our surprise, in several different model systems in mice, we've also found that enveloped AAV is more potent or more efficient than conventional AAV. This was a study with Federica Mengozzi for hemophilia B, and we showed that enveloped AAV encoding factor IX was much more potent than standard AAV. And for redosing, we also think that enveloped AAV has a potential to be used for redosing to engineer the membrane with certain ligands, and that's currently under development. So we think we have a lot of potential and opportunities with this platform based on what we've done. One of the first things is that we have strong IP. We have an issued patent for enveloped AAV, which has broad composition claims. We've shown in several different tissue targets and organs that in mice that enveloped AAV outperforms AAV, including the brain, the liver, muscle, the eye, and also the cochlea of the inner ear. And with some great collaborators, we've also shown that enveloped AAV shows therapeutic benefit when you encode some certain transgenes. We've shown that in Alzheimer's disease, hemophilia, and also hereditary deafness. And I should also just quickly mention that we view enveloped AAV as a matrix platform, and that you can engineer the vesicle in many different ways, and then combine all those engineered vesicles with any given capsid you'd like. So some current developments. What do we need to take enveloped AAV to the next level towards commercialization and finally to use in patients? Right now, we're testing scalable purification approaches for enveloped AAV. We're also testing scalable production in suspension cells. Another really critical part of our study is to find out if we can translate our studies in mice to non-human primates in the ability of AAV to perform the same way in larger animal models to make sure it has a really good chance to translate to humans. Once we do those studies, we'll be able to pick our main indications to whatever organ tissue we can, and then we can map a path towards the clinic. So with that, I really want to thank you for your attention. I hope I've given you an idea about the potential of enveloped AAV, and thank you very much.